So the sarcomeres are nothing but the structural and the functional units of muscle contraction within the muscle cells. Now, if this is a muscle cell, let us call it as a muscle fiber. There are numerous parallelly arranged cylindrical structures which are present within the cytoplasm of the muscle cell. We had discussed that these cylindrical fibers are referred to as the myofibrils. Now on these myofibrils you will find these repeating units which are marked by these cross striations which are referred to as the Z line. So this is a Z line, what you can see over here. So one Z line to the next Z line. So which is the next Z line over here? Now let me consider this to be the second Z line in the immediate vicinity. So this will be the second Z line over here, the blue line. So there is one Z line here, there is one Z line over the other side. Together, the space between the two Z lines constitutes the structural and the functional unit of muscle contraction within the myofibril. See, this is the myofibril, this tiny cylindrical structures present within the cytoplasm of the muscle fiber. And you can see that there are two types of filaments within the sarcomere. The blue colored filaments called the thin filaments which are projecting to the center from the Z line, and then you have the red colored filaments which are referred to as the thick filaments. The thick filaments are made up of a protein called myosin. They occupy a central position in the sarcomere. The thin filaments are made up of a protein that are referred to as actin. They are projecting from the Z line. They are attached to the Z line on both the sides. And in the center, you have another elastic membrane that is called the M line, which is keeping all the the thick filaments or the myosin filaments in place. So this is how the sarcomere actually looks under the microscope. Can you see that there is a darker zone and a lighter zone? The darker zone is called as the anisotropic band and the lighter zone is referred to as the isotropic band. Why does the central region of the sarcomere appear dark? That is because there is both actin and myosin. And if you identify this zone where there is only myosin, only the blue protein filaments are represented in the diagram, where there is no myosin, only actin. That region where there is only actin is referred to as the isotropic band, which looks light under the microscope, whereas the central zone, which appears dark under the microscope, is referred to as the A band or the anisotropic band.